Hello? Can you hear me? Good. Well, good morning, everybody, and welcome to our annual BioForo meeting. My name is Ana Zubiaga, and together with Alicia Alonso, Professor Alicia Alonso, who is at the left, the further on the left, we are the BioForo organizers. And today we are accompanied by Professor Fernando Plazaola, who is the Dean of our school, and Professor Immaculada Arostegui, who is the Vice Chancellor of Research. And they are the ones who are going to open today's event. Fernando Plazaola will offer some opening remarks, followed by Immaculada Arostegui, and after that we will start with the meeting. So, Fernando.
Thank you very much. Bueno, hasiko naiz Euskal Herriko Unibertsitateko ikerketaren arloko errektore ordea, BioForo ekitaldiaren antolatzaileak, zu zeu, Ana Zubiaga eta zu Alicia Alonso. Lehenik eta behin BioForo ekitaldiaren antolatzaileak eskertu nahi ditut eta eskertzen ditut, irekiera ekitaldi honetara konbidatzeagatik. Eta izan ere oso pozik nengoke beste urteetan egin dudan bezala, goizeko hitzaldi guztia jarraitzen, baina aurten ezin dut, azkeneko momentuan lan desberdina sortu dira, eta utzi egin beharko zaituz eta bukatu eta zeitxo. Beraz, egunon guztioi eta Zientzia eta Teknologia fakultateren aldetik, ongi etorriak zarete, urtero Paraninfo honetan ospatzen den BioForo ekitaldira. In this foro, researchers that have performed very major developments in biosciences during the last years explain them to bioscience students of our faculty. In fact, it is very enriching for our students because it allows them to get awareness of the state of the art of the field in which students can introduce themselves into research. This year, the topic of this edition is breakthroughs that are rocking the biosciences, in which several prominent scientists who have made very important developments in recent years will speak to you, Bioscience undergraduate and graduate students of our faculty, about their studies and the recent developments that contributed to these advances that are shaking biosciences. Therefore, I thank professors who have come to the BioForo bed, for sharing their knowledge and their research work. Indeed, I thank Guto Riz from Cardiff University, Purificación López García from CNRS University Paris-Saclay, Asier Gómez Olivencia, Ikerbas researchers at this Faculty of Science and Technology, and Luc Cole from the Deseus Mujer Foundation, Barcelona. Well, I have been heard just now that he is not with us, unfortunately, because of the COVID. But anyway, I have heard also that you will have the opportunity of listening to his talk in the near future online. No? Okay. So I want to thank for all of you for allowing our students to get to know the developments you have achieved. Before finishing, I have to say I am very pleased for the great job the organizers of BioForo, professors of our faculty, are doing year after year in order you, students of biosciences of our faculty, to get informed about the state of the art in different research fields of biosciences, and to allow you learning how to proceed after finishing the degree or the master degree. Eskerrik asko Alicia eta eskerrik asko Ana. Enjoy the BioForo meeting and learn a lot.
Thank you. And now, Professor Immaculada Arostegui.
Zientzia eta Teknologia fakultateko dekano jauna, BioForo jardunaldiaren antolatzaileak, Ana eta Alicia, ikerlariok, ikasleok, egunon eta ongi etorriak 2022ko BioForo jardunaldi honetara. Lehenik eta behin, nik ere eskerrak eman nahi dizkizuet antolatzaileei jardunaldiaren irekiera ekitaldira gonbidatzeagatik. Bizkaiko kampuseko errektore ordea zen etortzeko, eta bere faltan niri luzatu didazue gonbitea. Eta egia esan plazer bat da niretzat zuokin tartetxo hau partekatzea nire fakultatean. Baita nik ere gustoko izango nuke goiz osoa hemen geratzia, baina beste ardura batzu direla eta alde egin beharko dut. Baita Euskal Herriko Unibertsitatean ekimen honetarako laguntza ematen duten erakundeak ezin ditugu ahaztu, alde batetik Zientzia eta Teknologia Fakultatea, Bizkaiko kampuseko erretoreordetza bera eta Biofisika Institutua ere eskertzen ditugu. Good morning and my warmest welcome to this BioForo workshop. First of all, I would like to thank especially to the organizers of the event, Ana Zubiaga, Alicia Alonso. I know that you have been in charge of this workshop for a long time, actually around 15 years. A large number of speakers have participated year after year in this workshop and lecture series around BioForo, both local and international, covering a wide range of topics and showing a picture of the different areas in which the Faculty of Science and Technology works, but always with a common focus on biosciences. As a member of academia, I know how difficult it is to maintain an activity active for such a long time. And you did it, not just for the annual meeting, but also for the seminar series during the academic year. Therefore, I would like to express my warmest gratitude and congratulations for this. Secondly, I would like to welcome the speakers of this 2022 edition for your participation. You come from different European countries and accept to share your research with us. So thank you so much to all of you for your contribution to this complementary training for students and researchers at the University of the Basque Country. The topic selected for this edition, based on what the prestigious journal Science was considered as the most significant findings at present, such as intelligent protein design, evolution of archaea, embryo culture, and microevolution of Neanderthals, is very suggestive. I'm sure that the contents would be very interesting as well. And last but not least, I would like to welcome all the participants, researchers, and especially students, bioscience students of the faculty. You are the main focus of this activity. It wouldn't exist without your participation. I just said a moment ago that it's difficult to maintain an activity of these characteristics over time active from an organizational perspective. However, what is clear to me is that from a participation point of view, it's even much more difficult to get a good rate of attendees. Then thank you very much for your attendance. Felicidades a todas las personas participantes por el éxito que habéis obtenido con esta iniciativa del BioForo año tras año. Espero que siga vivo durante muchos años más. Ana, Alicia, que alguien coja vuestro testigo en la organización, de lo cual estoy segura conociendo el equipo que habéis creado a vuestro alrededor las dos. Pero sobre todo espero que la aceptación y participación entre el estudiantado siga siendo tan alta. Creo que es, de verdad, la clave del éxito. Zorionak, benetan, eskerrik asko eta goza ezazue, jardunaldia segun osoan zehar.
Well, thank you very much, Fernando and Inma, for these very warm words. As they mentioned, they are very busy, so we're going to let them go to do their work, and we are going to start with the event. Thank you very much for having come here.
Thank you to you.
Now it works. Now, before you come, I'm going to give a little presentation. Is that okay? Yeah, so let's start with giving you a few details about what BioForo is. Let's see if it works. Right. So Inma already mentioned, and also Fernando already mentioned a little bit about the BioForo. The BioForo has been going on for 15 years. We started in December of 2008. And during this time, we have been running 2 types of activities, both Alicia, Alonso, and myself. One of them is the annual BioForo meeting, which we do every December, and then also seminars, usually 1 each month or so. So that altogether is about 15 meetings and over 200 seminars. The total number of speakers up to now is over 250, and the total number of attendants would be approximately 12,000. It's about 800 per year or so. And the speakers are mostly local and from other Spanish regions. As you can see, pretty much half of it is about the autonomous community, the Basque Country, and other areas of Spain. Also, a big chunk of them come from the European Union, but we do get from time to time speakers from other countries such as the US, Canada, the UK now. That's not in the European Union anymore. We should put them outside of that. Anyway, so we would not have been able to run this activity if we didn't have any financial support. And we would like to thank today to our main supporters, the Vicerrectorado del Campus, Gorka Moreno, who couldn't come today. Instead, Inmaculada Arostegui, the Vicerrectora de Research, came. Fernando Plazaola, who is the Dean of our school. Probably all of you know him. He seems very interesting there. But also Asier Fullaondo, who is the coordinator of the master's program in molecular biology and biomedicine. Besides those, we also get the help of the Instituto Biofísica, the Iker Basque Foundation, the Basque government. So from here, I would like to thank to all of them. Thank you very much. Let's give them an applause. Oops, wait. Now, there's something going on here. Oh, okay. So the topic of today is breakthroughs that are rocking the biosciences. And why is that? Why did we choose this topic? You probably know that Science magazine, the scientific magazine, at the end of the year, in fact, the last issue of the year, they publish what they think is the major breakthrough, the major discovery of that whole year. And so that's what the last issues of this year's look like. You know, in 2011, it was the HIV treatment. I don't know if I can see well. Then came the Higgs boson, later cancer immunotherapy, the Rosetta mission, CRISPR in 2015. Then it was this collision of 2 stars in 2017. 2020, of course, was the vaccines. And in 2021 was prediction of proteins using artificial intelligence. So, in fact, this year, as I mentioned, this is the major breakthrough that Science magazine has thought about, the prediction of the proteins. As you well know, for 50 years or so, we have been mapping the structure of proteins using mostly X-ray crystallography, then came cryo-EM. But the last 2 or 3 years, artificial intelligence has made its way in. And several software programs have been developed which are helping predict the structure by the thousands of proteins. So the whole game has changed all of a sudden. It is helping, for instance, in defining de novo proteins, like we will hear today. But it's helping in many other ways, some of which we still haven't figured out. But in the next several years, we will definitely see a new era opening in the area of biology and medicine by using this prediction of the proteins. So we will hear more about that with the first speaker. But then, besides the breakthrough of the year, there are 10 runners-up every year. That is, 10 other major discoveries that have been made during that year maybe didn't make it to number 1, but they are also very important. And one of them has been this one, a missing link microbe emerges. That's what the title was of the runner-up. And that's because, as you probably know, the microbiologists have been debating for many, many years, they still are, what the origin of eukaryotes is. And very recently, some scientists have been able to culture a kind of microbe. It's called Asgard archaea, and they have been able to sequence the DNA. And what they found is that this archaea carries some eukaryotic genes. And from there, and many other results, they have confirmed, more or less, they think they've proven, that archaeas, or this Asgard archaea, or some other more primitive archaeas, were the predecessors of the eukaryotic cells. So we are very lucky that we have an expert in archaeas and microbe evolution with us, so she will tell us all about it a bit later. And now, continuing with evolution, another runner-up has been this one. Ancient soil DNA comes of age. As you well know, in the past several years, a lot of ancient DNA has been analyzed from fossils, both humans and also other animals and plants. And that is allowing us to understand how these species evolved, how they interacted, whether there have been any mixing among them, all sorts of things that are allowing us migrations. We're learning about that, things that we thought it would be impossible up until very recently. However, there aren't that many fossils of humans and other species. So very recently, a new technological development has been accomplished, and that is to use soil from which to extract DNA, and from that DNA analyze what's going on in that cave or in that ecosystem. And that has been a huge development because it's allowing us to understand many things that we would not have been able to understand if we were only relying on fossils because of the scarcity of these fossils. And I'm very proud to tell you that the members of the geology department of the University of the Basque Country have been pioneers in the development of this technique, working with people in Atapuerca Cave. And we're very lucky that one of them, Asier Gomez Olivencia, is with us to tell us about his discoveries. I would also like to mention at this point Arantza Aramburu, also a professor of this university in the geology department, who has been instrumental in the development of this new technique. And the last one, the last runner-up, is this one: embryo husbandry opens windows into early development. And as you will know, what happens in early development is important, is relevant for understanding why miscarriages are occurring, what different genetic mutations can make into, or can develop into problems later in life. It's also very important for the improvement of in vitro fertilization, but it's very difficult to analyze this very early development, first because of technical issues, but also because of the ethical and legal issues of using human embryos. So in the past year or 2 years, new developments have been made. For instance, some scientists have been able to develop mouse embryos for up to 11 days in a jar like this. This is a little mouse embryo. And 11 days for a mouse development is half of its developmental period. So we're able to see half of the development in a jar for a mouse. Also, other developments have been the blastocyst, the analysis of blastocyst in a jar like that by using IPS, induced pluripotent stem cells. So we avoid the fusion of the sperm and the egg, which is ethically a problem, and we have been able to use IPS for this analysis. And this is helping a lot, like I said, to understand why problems occur during the development, early development, but also is helping embryologists and people who work in clinics try to improve their in vitro fertilization techniques. So our last speaker is an embryologist who works with techniques such as these or others that they have developed. Unfortunately, as the dean mentioned, he just got COVID, and quite a nasty COVID. So he has not been able to come today. What we will do instead will be next week, we will arrange an online seminar for you. And so I will send you in an email, I will send you the link to which you guys can go, and whoever is interested can follow the seminar of the last speaker, okay? So this is the plan. We have the plan, now we're going to start with the first speaker. And the first speaker is Geto Riz. And for that, I have asked Aitor Hierro. Some of you may know Aitor Hierro. Aitor Hierro is a scientist, a researcher in CIC Biogune, who is doing groundwork, really interesting work. We have invited him a couple of times already that I know of to the BioForo seminars to tell us about her really interesting work. But today he's not going to talk to us about his work. He's going to introduce Geto Riz. Thank you, Aitor. Let me see. Oops, this is mine. I have to give it to you now. Sorry.
Okay. Yeah. Okay, thank you all for coming, and hopefully you enjoy all this seminar series. As Anna mentioned, I was here like a few times giving presentations at this forum, and today is a pleasure to have the opportunity to present one of the speakers, who is Geto Riz. He's a very talented young scientist who has been recently appointed as assistant professor at the Cardiff University. Previously, he worked in two of the best recognized labs in protein chemistry, protein design, and synthetic biology. In one lab with Professor Dec Wolfons at the University of Bristol, and in the other lab with Professor Bert Hocker at Beirut University in Germany. So during this time, he focused on aspects of the protein folding problem and the application of this knowledge into new design of novel proteins. Dr. Geto's current research combines synthetic chemistry, molecular biology, structural biology, and computational protein design for new applications, as for example, what he is going to talk today for delivery compounds intracellularly. So, Geto.
Okay. Good morning, everybody. It's nice to see all of you. There's lots of people here, I guess, undergraduates, graduates, and it's really nice to see it so full with lots of eager faces. By all means, at the end, ask questions, but if you're uncomfortable, then please just come see me afterwards to have a chat. I really believe there's no such thing as a stupid question, so if you have any ideas or something, come see me and we'll talk. Okay, so thank you very much for the kind introduction that I've had now by numerous people. I'm Gitta. I started in Cardiff University in the UK about 3-4 months ago now, where I started, where I'm going to have my own research group. And I'm based in Cardiff, and Cardiff is in the UK. So this, if you look on the right here, you'll see the UK that's in the kind of bluish colour. And then Wales is this section here. And I'm in Cardiff, which is the capital of Wales here at the bottom. And it's about 2 hours latitudinally, from London. And it has a lot of similarity, I think, to Basque Country as well. There's around 3 million people, just like you have here. We have our own language, which is quite old, not quite as old as your language, but it's still quite old, so like, 1,400 years old or something. Yeah, and it's a lovely city that's just on the coast. So I've been asked to talk today about what I'm interested in and what we do, and I'm within this field that's been developing really more recently at a rapid pace, which is protein design. And what is protein design? Well, the idea is we spoke briefly before about how we can now use computational algorithms maybe to start predicting what protein structures look like. But really, biology is fascinating and there's a lot there, but there's so much that biology has never explored, right? So if we think about evolution, it's been going for, hundreds of millions of billions of years, but there's a lot that nature still has never explored before, lots of protein sequences and DNA and RNA that's just never been explored. And so, well, why would we want to do that? Well, there's so many interesting things that we can do now that we can start to design new sequences. And the idea of designing is essentially that we can take any sequence that we want, we can predict what the structure should be, and then we can apply it towards totally new applications. And one of the research areas I'll be going into, developing from my postdoctoral work, is this idea, can we design new enzymes to catalyze reactions that nature just doesn't do? And so what you'll see at the top here is something I've been trying to develop, and this is a chemical reaction that chemists use a lot in the lab, but there's no biological equivalent. There's no enzyme out there that can do this. And so can we design totally new enzymes to catalyze reactions that nature doesn't do? Why would you want to do that? Well, if we can use proteins, we can make it better for the environment, we can run it in water, and so on and so forth. And also, it has exquisite selectivity that you can get with proteins to design new small molecules. But today, I'm going to talk about a project of designing much, much smaller sequences, so just peptide sequences. And this is building a lot on what I did during my PhD, and subsequently when I moved to Germany to do my postdoc. While I was there, I started up this new collaboration with my old PhD supervisor in the UK. And it was building on a lot of knowledge that I'd learned about these peptides, these type of peptides called coils. And what these are 2 or more alpha helices that can wrap around each other. And you can make all sorts of structures that have never existed in nature. And so we've got these kind of things here where these peptides come together to make protein-like structures. And then we can look at the dynamics of what they form. We can design totally new structures. But what I'm going to focus on today is, okay, it's cool that you can make these new structures, but how do you apply them? What can you do with these things? And so today, my talk is going to be focused on this last part. So designing peptides for cellular delivery and subcellular localization. And the idea here is quite simple. Can we design something that's totally orthogonal, so totally separate to biology, and then add it into a cell to do something very specific? And so the concept here is, can we take one pair of peptides, so one here in blue and one in red, and the idea is, can we design 2 sequences of peptides where one doesn't interact by itself, but only when it finds its partner peptide do they come together and interact. Now, can we take that a step further? Can we take one of the pair here, and can we have it delivered exogenously to a cell? so that it can make it into the cell and find the transiently expressed partner and bind to it. And then the last step, can we take it even further? Could we then, for example, deliver something else into the cell? So we take something that gets into the cell, it gets delivered, and then it has some kind of function once it's in the cell. Okay, so how do we go about doing that? Well, first we're going to have to design our peptide sequences. And in order to do that, what we did is it took a team to put together to do it. And so this was a true collaboration where I had, in addition, Jessica Cross and Will Dawson. Will Dawson did a lot of the peptide work in Bristol in the UK. And also Jessica Cross, who's a fantastic scientist. And all the images that I'm showing of cells today was all her work. And she did an incredible job of applying this new system we developed in cells. Okay, so how do we design these sequences? Well, with all the technologies that are available at the moment, what we actually ended up doing for this was very simple. We looked at natural sequences to begin with. But in order to do that, you need to think about the type of sequences you have and what the peptides are. And so this diagram we have here is a representation of one of the helices, and this is the second one here. And I'm not going to go into a lot of detail of the sequence design today, but if you just understand this, that what we have are these interacting parts between the two helices, here on the left and the right, and then these blue parts are the parts that will be pointing out towards the solvent. And what you can then do is you need to know that with these types of sequences, they repeat every 7 amino acids, these coils. And so you can repeat the sequence as many number of times you want, and it'll just make longer coils that interact. And as an example, you've probably seen coils everywhere, but you're unaware. They happen naturally. There's a lot of them. There's some that's involved with cytoskeleton, so like quite large ones that are rigid. Some of them are transcription factors that bind to DNA. And even when we talked about COVID and we were talking about the spike protein, well, the spike was actually to some extent caused by the coil that was like spiking out of the structure. Okay, so what do? We then learn from all the natural sequences in order to make a new one. And so what I did was I took information of all crystal structures of known natural sequences. And what we can do then is we can look to see to some extent like a consensus sequence, so the type of most frequently seen amino acids at different positions in these coils. And then based on that information, we can learn that what we want is to put these leucines and isoleucines that are buried interacting with these two, between these two peptides. We need to learn a lot more to do the rest, and so we then can look at more interesting things. So we can say, what are the different amino acids it's interacting with on the other helix? And based on that information, we can then start to fill out the rest of the helix of what we're going to design. Then the next step is we also need to think about what's going to be on the outside of the system. And here we just need to put things that make it very stable. We're going to need to design it to have some kind of chromophore so we can see. So we put tryptophan amino acid in there and other things to help solubilize it, like the glutamines. And then crucially, what we found, and this is what people in the field of protein design are finding, is you can design in specificity, so a way that things interact, be it a protein with another protein or a peptide with a peptide, by introducing polar residues that are buried at the interface. And key to this was realizing that we can design in a buried arginine into our system. And that gave us then the specificity it needs to find each other and also lock in the right place to make the interaction. Okay, so we have all the information we need, so let's now put it together and design sequences. So one of the things I did was generate some computational models of what these things would look like, but ultimately what we got are these sequences. And so what we have here at the top is what I'd just call a homomer. So this is a single peptide that should interact with itself. So one peptide will find another one of the same one, and it should form a dimer. And what you can see is here the sequence from N to C, and we can chemically synthesize these things just using essentially like robotics. We use a machine that can chemically synthesize this peptide to test it out. And what we saw is that when you put it into a phosphate buffered saline solution, we get something very helical. So we know that once it's in solution, it's forming something very helical. And what we can see here is this circular dichroism spectrum. And on the right, we see that we have our two peaks here. So we have Here on the left are 208 nanometers, and then at 222. And this is, if you're not aware, just very typical of something that's very helical. So it tells us we've got something helical. And the amazing thing is, this is only 30 amino acids long, but when you heat this thing up, it's pretty stable for such a small peptide, right? So you have to go up to about 63 degrees in order to melt this thing. So for such a small sequence, because we've designed it so accurately, it means that it's very stable up to a high temperature. Okay, but what we really need for this to work is we need two different peptides, right? And so what we then did was desymmetrize the system. So we start with something like this, and then we just take this apart and design slightly different sequences so that they don't interact by themselves, and only when they come together will they form a structure. And so what we have here, first of all, is a similar sequence, but we've changed it now so it's very negatively charged, right? It's got lots of these glutamates there. And the cool thing is when you put that into solution now, even though the sequences are very similar, if you compare them, we find that it's just really unfolded. It's just forming a random coil, so there's no interactions happening, it's not helical. And then we design another one, a second pair to go with it. And that's this positively charged one. And again, we see the same kind of thing, right? It's just very much unfolded by itself. And then we do the key experiment where we take our one peptide, peptide A that's negatively charged, and peptide B that's positively charged, and we put them together and we mix them. And what we see is really cool. We get something that's even more helical than this single peptide at the beginning, and also it's extremely stable, right? And then we're able to confirm using analytical ultracentrifugation that this thing really is forming a dimer in solution. So it's not lots of peptides coming together. We're getting a 1-to-1 ratio here of the two, as you can see from here. But coming from a background in chemistry and really enjoying structural biology, I wanted to have some more information, and we wanted to be sure that this thing really is what it is. And so one of the experiments we did was this labeling experiment. And I'm not going to go into a lot of detail here, but what we did is we covalently attach a fluorophore at one end of the peptide and then a fluorescent quencher at the other side. And so we can learn about the orientation of the two peptides relative to each other, right? So the idea is if you label one side at the top with a fluorescent dye, and then we have a quencher at the bottom, if these interact as we designed in an anti-parallel way, when they flip and come next to each other, one dye should quench one dye should be quenched by the fluorescence quencher. And that's exactly what we see in this experiment. So we can tell in solution, in vitro at least, that what we have here is something that is anti-parallel. And in addition to that, another scientist within this team, Harry Thompson, he also developed a split assay to check that this is true also in the cytoplasm of E. coli. And the same we saw there, that it had a strong preference to interact in this anti-parallel fashion. And lastly, and maybe for me, one of the most interesting things, we were able to validate our structure. And what we were able to get in both cases were high-resolution crystal structures of these peptides interacting exactly as we designed them. Cool. So at this point, we have our structures. We have all the information we need, and we have a really robustly designed peptide. So I guess it's time to do some kind of cool biology with it, right? Okay. And this is where Jess got involved in the project. So now, can we use it as a new delivery system that's orthogonal to biology? And so Jess simply asked this question to begin with. She took the two peptides by themselves, peptide A and peptide B, and she added a red dye to them, so Tamra, that fluoresces. And she asked the question, can they both get into cells? And what we had was, obviously, one of the peptides is now very positively charged, right? And this is a classic, what you see in cell-penetrating peptides. And what we see here is we have at the top our peptide B, the positively charged one, and then underneath our peptide A, which is negatively charged. And what we find is once we've incubated the cells, these HeLa cells, with the two peptides, we find after we've washed them that only this positively charged one gets in, and it gets in very efficiently, not only to the cytoplasm, but also makes its way into the nucleus. So cool, we've got a peptide that we can deliver into cells. But the second question we need to ask is, if it can get into cells, can it then find its partner, bind to it, right? And so what Jess did in this case was she did an immunoprecipitation assay or a pull-down assay. And what she had was green fluorescent protein attached here to the negatively charged peptide. And what she did was then have it transiently expressing in the cells. And what you could show in this experiment is that we only see the red fluorescence from pull-down of peptide B in the case where we have everything together, right? So we have transient expression of this in the cell, we add this, it gets into the cell, binds, and then through the pull-down of GFP, we get this red fluorescence. And we see that only when we have all of the combinations of the right things in place that we see this fluorescence. So none of the controls do. Okay, great. So at this point, we've shown that we can get this one peptide to go into cell and it can bind its partner. But the story is all about, can we then do something more interesting now it's orthogonal? Can we get it into the cell and can it get to a subcellular location? And so this experiment is one of the key discoveries that we made in this project. So let me talk you through it a bit slower. So what we see here on the left, if you look at the diagram, is what Jess did was she introduced a transiently expressing version of a natural protein that's in the cell. And that natural protein is this LAMP1, which is the lysosomal associated membrane protein 1. And this protein is naturally embedded in lysosomes. And what she did was make a variant that had this GFP attached. So now, when we look at a green channel in our microscope, then what we're going to see is all of these lysosomes should light up green, right? And what we wanted to find out was then, if we introduce our peptide B to the cells, do we then see it go and find these green puncta, and then we see co-localization where they bind of this red fluorescent dye. And so what we see at the top here is this example where we have the GFP, but it doesn't have its partner peptide. And what we see when we put the two channels on top of each other overlaid, we see this like perinuclear clustering of all of the lysosomes. But we just see that when the peptide B gets in, it's very diffuse, right? It's spread out all over the cytoplasm and in the nucleus. And so we don't actually see localization because there's no peptide A here. And then the dramatic thing that then happens when we introduce our peptide into the system, here with APCC dye A, is that we now see this co-localization. So what we're getting is the peptide makes it into the cell, we get an interaction between the two, and we see co-localization where now the two peptides have found each other. Now, some of you may ask, well, is this just coincidence that they're both making them into the lysosome at the same time? And so Jess took some time to develop another system to show that you can really target other subcellular compartments. And what she did in this system was also show that if you move the peptide over to the mitochondria, you can get the same peptide to go in, and now instead of targeting the lysosomes, it's now recruited and binding to the mitochondria in the cell. Cool. So we've been able to deliver a peptide. Great. You know, it's a cool concept. We've been able to design it. But is that useful? Can you do something cool with it? And at the time, Jess was developing this different system, right? So she was learning a lot about networking within the cells, or network trafficking. And what she was particularly interested in were these kinesins, which is a motor protein within the cell. And what it does along these microtubules that you find throughout the cell is it helps network these vesicles around the cell. And specifically in our cases, we're interested in these lysosomes. And so it's an important protein that we have in the cell, and it has a wide range of functions, and also its dysregulation causes some human diseases. And what Jess had shown in a previous project is that she could take natural sequences that bind to kinesin, specifically to its TPR domain, and she made a very high affinity version by combining these two natural peptides which she called KinTag. And what that then does is this peptide combined to the kinesin, it then activates it, and then it causes anything that's covalently bound to this KinTag to be transported along the microtubules using kinesin. Okay, so the question is then, can we take this KinTag and deliver it exogenously to the cell using our new cell-penetrating peptide? And so the grand scheme, as I showed before, is this one, where we have on the right our peptide B, which is now fused to the kinesin tag, this kin tag. What we want to do then is be able to deliver that into the cell, and what we will have is some transiently expressing protein here, which has our peptide B attached to it, our peptide A attached to it. And what we want is the two interactions to happen. We want the peptide to find its counterpart, which is now on this lysosome. And then we want the kin tag to find its kinesin that's up here. What that should do then is it should recruit it and then strongly activate it, such that what happens is we see all of these lysosomes in the cell being trafficked right from the nucleus to the ends of the cell, to the periphery, if it's working properly. And so this is the experiment. When we look at the first row at the top, what we see here is simply, again, our lysosomally associated membrane protein, LAMP1, that has GFP on them. So they're all lighting up green in our cells. And what we can see is we have this perinuclear clustering again. So all these lysosomes are basically just all around the nucleus to some extent. The next control that we did to show was that if we then removed this dia from the system and then exogenously add our peptide B with kin tag and the fluorophore, we see essentially that the peptide can make it into the cell. It lights up everywhere in the cell and in the nucleus, but we don't get an interaction. So these lysosomes and late endosomes are essentially, the majority are still perinuclear. And now the grand experiment, can we take this totally new design peptide, introduce it into the cell, get the interaction we want, and then see this delivery and then activation of the lysosomal kinesin, and then see the transport to the cell periphery. And so what we have here in the grand experiment is this schematic here, and this is what we saw, which was quite cool. So if you look here, what we have, in fact, is co-localization. So we know that the GFP has found, so we know the green puncture from where the peptide is that's being transiently expressed, and we know from the red channel where we have our exogenously labeled peptide. And what we see is complete, essentially, co-localization. So the peptides finding each other. But more striking than that, what we can see is that we're getting strong activation to the point where the cell morphology is changing. These cells are still viable, they're still alive, but what's happening is because our design system is so strong, I guess the affinities, we're seeing that essentially the majority of these lysosomes have all been trafficked to the ends of the cells, so much so that it's starting to cause this weird difference, change in the cell membrane. Okay, so we've taken something from a proof of concept all the way through to showing how it can be used in, at least in one case here, using our kin tag. And the one thing to note is that you can also quantify this and gested this by counting the number of lysosomes and where they were distributed. And it really is, I mean, you can tell from the image, right? But it really is statistically significant. You can see these proportion of lysosomes that have been trafficked to the cell periphery. Okay, so just to summarize, the take-home message here is that we can now do some really cool stuff. I've only described here a small peptide system, but you can imagine that you could design much bigger proteins for anything that you want in the cell, or even, not just single domains. We can start thinking about designing totally new networks within cells using protein design, and they would be totally orthogonal, and we can do some very cool stuff in applying it to, new biologics, new enzymes. If you've got an idea, then there's an opportunity there. So what we did here is we took some bioinformatic information to design new peptide sequences. Based on that, we were able to deliver a robustly characterized sequence, doing it all in vitro, to prove that we know what's going on inside the cell on an atomistic level. We then demonstrated that one of the peptides could get into the cell, and then also that it could bind its partner. And lastly, I think most cool aspect of this is what we're now starting to see across protein design in general is that It's not just a crazy idea now of just designing sequences to see what they look like. We can apply them in real context and do some really cool stuff. And in our case, what we were able to show is we can deliver an exogenous peptide and have it show function within the cell. So that leaves me to thank everyone that was involved in this project. It was a real, true collaboration between me in Germany and some of the people in the UK. So Jess Cross, who did all the imaging, Will Dawson, who helped coordinate a lot of it in the UK while I was in Germany, Harry Thompson, who did the split assay in bacterial cells, Suraban Shanmugaratnam, who helped me a lot with the crystallography work, and then the 4 PIs that were involved, which included Professor Dec Wolfson at the University of Bristol in the UK, in addition to Mark Dodding and Nigel Savory there, and also to Berta Herker, who was my postdoctoral supervisor. So I guess to some extent with that, it leaves me to thank the 2 organizers who've put on this, what really looks like a really exciting meeting that's going on at the moment. So thank you very much, Ana Zubiaga and also Alicia Alonso. And more than anything, really, I'm so grateful that you all come along. And like I said, if you've got any questions, please come see me at the end if you feel uncomfortable asking now or you want to learn more about protein design. But with that, I think I should say thank you very much for listening. Esceri Gasco.
Bye bye. Wait. Bye.
Okay, thank you. Really beautiful talk. Do you have any questions? Anyone has questions? Well, in the meantime, while you are thinking on your questions, I'm going to ask one. Have you calculated the affinity or the yield of when this interaction between the 2 peptides happened? Is a really high yield when you incubate them?
Yeah, so you're talking specifically about the designed coil interaction?
Yes.
Yeah. So we ended up using the circular dichroism. There's a technique you can use with that to look at affinities, the melting temperature of various concentrations. And from that, we sadly, the affinity is too high using that method. So we know that it's below micromolar. Okay. And we can only predict, based on other systems we have, it's likely to be around like a single-digit nanomolar affinity that these 2 peptides have. So I think it would be cool to confirm it using a different technique. The one thing I would say, what's really cool about design systems like this is that depending on application, we can, very rapidly, we can design new systems that have lower or higher affinity. And one of the easiest ways to do it is we could just extend the helices. Mm-hmm. And then, these peptides, there's another academic in Germany now. She's shown that, Franziska Thomas, she's shown that you can basically extend or contract the size of them to get all the way from, sub-nanomolar, we're talking picomolar, all the way through to millimolar, depending on what affinity you want for your system. And I agree, I think we were very lucky. We struck a balance here, because we didn't want something that was maybe too high affinity to begin with. But yeah, we could easily design it, but it's probably nanomolar.
Really cool. And a second question regarding this positive peptide, the one that you just throw into the cells and it translocates inside. Can it carry really different things like nanogold particles and still cross the membranes, for example? I'm thinking on labeling things inside the cell.
Exactly. I would be hesitant to say that it could get very large things through. I think the mode that it would do it by would be very different then. It would be cool to try, right? Just to see if you had loads of this peptide on the surface of something bigger, would it still be able to make into a cell? And I think that's a cool experiment to try. We haven't done it. The only thing I would say that's cool is this Kintag peptide, even though it's quite small, it's highly negatively charged. So by itself, it would never want to go into cells. Okay. And the cool thing is that Jess tried to use various known peptides that can do this. There's the classic HIV TAP peptide, and then there's an an R6 peptide with lots of arginines, and none of these, once you fuse them to the peptide, the kin tag, work to get them into the cell. So I think we've got a lot to learn about how these cell-penetrating peptides work, and I don't think we still fully understand it, but it is cool, yeah.
Really nice. Yeah.
Thank you, yeah.
So,
Great work, thank you.
Thank you.
So it looks like it has a lot of future in terms, for instance, of precision medicine, functionalizing these peptides. Now, I was wondering, knowing very little about protein structure, there are other structures, there are protein structures such as beta sheets. Would it be something interesting to try to use this? Is it, so should we, coil structures and alpha helices are the thing to use for this kind of things, or can someone start to think about some other structures that may be interesting for these things?
So are you talking particularly about this application of cell penetration or in general? No, so the reason we've used coils is that there's a long history of designing these. Like long before we had really cool computational algorithms, people have been doing rational design on coils because it was one of the first structures ever envisaged, really. It was the first one predicted to some extent by Watson and Crick originally, apart from DNA. It's a really robust system and it works really well. Lots of cell-penetrating peptides are helical, and they are also, they must form, these peptides probably do form some kind of helix when they're on the surface. But really, people now are not just designing these small helical things. People are designing really complicated structures using mixed topologies, beta, alpha. Dak Wolfson had a paper recently where they thought, can you just make a peptide that only forms from 310 helices, which are very rare in nature, and they just got a crystal structure of a peptide that self-associates with just 310 helices. And I mean, that's really abstract, and it kind of feels, it's cool for me, but maybe totally pointless for a lot of people. Like, why do you want a 310 helix? But the idea that you can make something that it never existed in nature and prove that it can be made means that really the limits are, well, I think we were only limiting ourselves, really, when it comes to design. And so, yeah, of course, you could use, and lots of people do use other types of structures. Yeah.
okay, yes.
Well, I hope I didn't fail to hear
That's all right.
this concept specifically. But I wondered what was the kinds of cells that were used throughout the experiment,
Yeah,
and if there would be any major issues with using that specific specific peptides with more specialized cells.
Yeah, it's a really good question. It's a great question. I didn't mention, these were just HeLa cells. We haven't tested it on other cell types for eukaryotic cell types apart from HeLa cells. They're very happy with this peptide. There's no, we don't see any signs of like loss of cell viability, but we've never applied it to other systems. Again, I think one of the cool things about designing peptides is that if you design it for a particular application, you don't have to use these exact peptides, right? And so what you could then do is, if you were working on, say you wanted some kind of like delivery therapeutic or something, whatever it is, you could then design a sequence that's tailored for that application. So it's cool that we can now take a sequence and just use it and try it, but we can always design a new one and it might be much better for your application. But yeah, great question. Thanks for asking.
Okay.
Thank you for your talk. We were thinking about if there is any possibility of using this approach to cancer cells, because maybe there is something that could be done with this peptide that you introduce into the cell, maybe even including in this sequence some specific peptide that is toxic or have any other negative effect. And maybe there is not something like a specific peptide in the cancer cells, but maybe yes, maybe we know that there is something that is in higher amounts than in the normal cells. So we were thinking about this, if there is something in the research nowadays.
Yeah, that's a really great question. That's an excellent question. And the reason it's a really good question, and I can't say a lot about it, is because I think my old PhD supervisor really liked this idea and has applied to get new funding to do things similar to that, which I can't talk about because there might be some patenting to do with it as well. But you know, you've just thought of something excellent there. But the idea is there, right? You don't have to even target two orthogonal things here. You could, like you said, go for something endogenous in the cell. We could design a new peptide here that's not going after its partner, but say some special peptide that might be in a cell that is not good for the body. I can't, that's all I can say, but it's a really good idea.
First of all, great work. And my doubt is, and instead you have said that you have used the anti-parallel coil.
Yeah.
But for example, if you try to do with parallel coil, it will be the same or not?
Yeah. So one thing I very much glossed over, that's a great question again, and it's something I just kind of sped over, is that I think it would work just as well if you used parallel or anti-parallel. There's nothing magical about anti-parallel. The reason I wanted to do it, again, is a very, academics are really stupid sometimes. They just want to do something because it's, ambitious or fun. And it's because no one had any crystal structures of designed anti-parallel ones. So I think it would work perfectly well. Like you said, if you could use a parallel or anti-parallel, you just fuse the things at whatever termini you want, right? But I thought it was a cool goal because they've existed for, we've known about their structures for over 50 years, but no one's been able to get a crystal structure of an anti-parallel design. And so just for me, for our small design community, I thought, yeah, let's try and do that. But you're absolutely right. It would work with parallel as well, yeah.
Okay, beautiful discussion. So if there are no more questions, we can thank again Gitos for his really nice talk.
Okay, I'm going to introduce you to the next speaker. Puri López García, while she's putting her microphone, let me introduce you to her. Puri López García is the research director for the Centre National de la Recherche Scientifique, that's a CNRS, in France. She founded and now leads the Diversity, Ecology and Evolution of Microbes team at the Ecology, Systematics and Evolution Unit of the Université Paris-Saclay. She got her PhD in Sciences, in biology, from the Autonomous University of Madrid. And then she went to, and the work was on the genome structure of halophilic archaea. Then she worked for several years, first as a postdoc, and later as an associate professor in various institutions, such as the Université Paris in France, Universidad Miguel Hernández, and Universidad de Alicante in Spain, also Université Pierre and Marie Curie in France. And then in 2002, she was offered a research position at the CNRS, up until now. Her main research interests lie in the area of microbial evolution and in early life history. She has a strong expertise in the study of microbial, bacterial, archaeal, protista, diversity, and ecology, using metabarcoding, metagenomic, and phylogenomic approaches in widely diverse ecosystems, which include also extreme ecosystems. Her research is highly interdisciplinary, notably at the interface between bio and geosciences. And she is recipient of many recognitions and awards. I would like to mention, for instance, that she is a fellow of the EMBO and the American Academy of Microbiology. She's a member of Academia Europea and an associated member of the Royal Academy of Sciences and Fine Arts of Belgium. She was recently awarded also the CNRS Silver Medal. So you get an idea of what Puri López, our next guest, is. And I welcome her, and I'll give her the floor is yours, Puri.
Thank you very much, Anna. I'm very glad that you hosted me here today, you and the rest of the organizers. So, egunon, good morning to everyone. And as you said, I'm working for the French Research Council, the CNRS, at the Université Paris-Saclay. This is close to Paris, but not quite Paris. It's actually a very nice forested area. So if you're interested, come and visit. And me and I and my team are particularly interested, as Anna mentioned, in the study of microbial diversity, ecology, and evolution at a very broad scale. And it is in this context that I'm going to talk about how the discovery of Ascararchaea and why have impacted our views on the origin of eukaryotes. So eukaryogenesis, the origin of the eukaryotic cell, was a major transition in individuality together with the origin of life itself and with the various independent origins of complex multicellularity with tissues, etc., animals, plants, fungi, kelp, and others. It was a unique event. It only happened once, and it led to a considerable average increase in cellular complexity. So how did this happen? At the beginning, early studies of the microbial world were done using microscopy, and then people already realized that there were two broad categories of morphologies in the microbial world, much simpler morphologies that corresponded to organisms that were called bacteria, and more complex organisms that usually had bigger cell size. It turned out in the first part of the 20th century that these two types of very broad morphologies were underlined by two different ultrastructures. The prokaryotic one, where you have the DNA directly immersed in the cytoplasm here, or another more complex on average cell structure where you had endomembrane systems defining a nuclear region containing the chromosomes, the genetic material, and here you have DNA replication and transcription occurring in the nucleus and translation in the cytoplasm. So it was logical at that time to think that eukaryotic cells evolved from simpler cells just by a process of complexification. And so this led to the very classical early view on the origin of eukaryotes, going from some type of Lucas, a last universal common ancestor, going to these simple cells, bacteria, and then from there to eukaryotes. So this linear view of evolution was later challenged, in particular in the 1970s, when we had the first attempts to build a universal phylogenetic tree of life based on molecular information, on molecular markers that were supposed to host information or to store information about evolutionary history. And this type of analysis of work was done with the aim of establishing natural classification where you could have in the same phylogenetic tree all known organisms, cellular organisms, from bacteria, coli, etc., to animals and plants. So this type of work was done by Carl Woese in the 1970s using ribosomal RNAs as universal markers, and yes, he demonstrated that we could build such type of trees, and he had a surprise that a particular group of organisms, bacteria-looking organisms that live in weird environments, very salty, very hot, etc., segregated and formed a third independent group that were called the Archaebacteria and later rebaptized in the domain Archaea. And in the first rooted trees, these Archaea were placed as the sister to eukaryotes. And so we have a problem here because we have two main type structures, ultrastructures, but we have three phylogenetic domains of life. So eukaryotes do not come in this linear form as thought previously. Where do they come from? And the first element of response came from the realization that some eukaryotic organelles derive from ancient symbiotic bacteria. So these ideas were popularized by Lee Margulis Sagan at the time, who somehow retrieved previous ideas by Ivan Walin and Konstantin Merezhkovsky, who had proposed that mitochondria and plastids derive from ancient endosymbiotic bacteria. And the final demonstration for that came more or less at in the late 70s again, when cyanobacteria and plastid markers placed together, showing that plastids derive from cyanobacteria and mitochondria plays a sister group to purpobacteria, alpha protobacteria, some of which are photosynthetic. So these had implications for the origin of eukaryotes, and the main idea that has prevailed until very recently is this one. Fortunately, it's a bit too light here, you cannot see, but here we have a last common ancestor. The idea was that eukaryotes derive from a third proto-eukaryotic lineage, sister to archaea, consistent with Calwos's tree, in which you have the evolution of all typical features of eukaryotes, but the mitochondrion. That means a complex cytoskeleton, endomembranes, the nucleus, and very importantly, the capacity to phagocytose. And with this capacity, the ancestors of mitochondria could have been incorporated in the cell. Nonetheless, already at the turn of the century, some people contested this view, and because actually we did not have a direct evidence for the existence of a third proto-eukaryotic lineage devoid of mitochondria. We already learned at that time that all extant eukaryotes have or have had mitochondria in the past. So there was not an evidence for such a pre-mitochondrial eukaryotic lineage. And hence, other models actually propose that there were only two major lineages and that eukaryotes result from the symbiosis of those. Among these models, we have the serial endosymbiotic theory by Lee Margulis, who also proposed that the ancestor, the host that acquired the ancestors of plastids and mitochondria was itself derived from a symbiosis between an archaeon, thermoplasma-like archaeon, and spirochetes that would have provided a motility. That part is not supported by current data, but yet the idea was there. And there were other models by the late 1990s that you have depicted here that also proposed that eukaryotes evolved by symbiogenesis. You have two of them, the hydrogen hypothesis and Xerxes model, that propose that eukaryotes derive from the endosymbiosis of the ancestor of mitochondria within an archaeon. And then the syntrophy hypothesis that I had the opportunity to postulate with David Moreira that involve a third partner. So we have the free chair nucleus would be an archaeon engulfed into delta proteobacterium, and the mitochondrion would have derived from a second endosymbiont. All these models have in common the proposal of metabolic interactions as the basis of the symbiosis, leading to the evolution of eukaryotes. Interspecies hydrogen transfer in the case of the hydrogen and the syntrophy hypothesis, and sulfur exchange in the case of the Xerxes models. But all these models at that time were kind of dismissed. People didn't really take them seriously. And the mainstream idea was that of a third proto-eukaryotic lineage. Things have changed radically today, thanks in part to our exploration of the microbial world. So we have a variety of tools and approaches with which we can explore the diversity of natural ecosystems, and in particular the diversity of microbes that cannot be, that are not amenable to culture, using metabarcoding approaches, so gene amplicon sequence. You have also metagenomic approaches where you sequence the genomes of the community and you can reconstruct metagenome assembled genomes from organisms that are not cultured. And you can also use single cell genomics. You can separate individually microbial cells, samplify their genome or their transcriptome, and then study those sequences. And with all these approaches, we have been populating the tree of life for the past 10, 20 years. And in particular, this is a more updated view of the tree of life. Many of these lineages with a red dot correspond actually to phylum level lineages for which we lack culture representatives in the lab. Most biological diversity is microbial and particular bacterial, so bacteria dominate the world. And you have archaea and eukaryotes here. And let us focus in this particular group that is called the Lochiarchaeota, and that is a member of this Ascararchaea that occupy us today. So Lochiarchaeota was a group already known by 16S ribosomal RNA sequences, but the first studies of genomes of these organisms were done by Anja Spank and co-workers, and who were studying metagenomes of sediments close to this Loki's Castle hydrothermal system in the Arctic mid-ocean ridge. And in these maps that they reconstructed, they observed the presence of several genes that were eukaryotic-like. And these were much more numerous in this archaea, Lochiarchaeota, than in the rest of archaea. And so she had the idea of including eukaryotes in her trees, and she noticed that eukaryotes placed as the sister group to this Lochiarchaeota using conserved universal markers. So of course this generated a lot of excitement in the scientific community, and there were many more MACs of relatives to this archaeal group that collectively are called the Ascararchaea in reference to the Nordic pantheon. So all these groups are named with God's names of this Asgard pantheon. Odinarchaeota, Thorarchaeota, Lochiarchaeota, and Heimdallarchaeota. At that time, there are many more now. Mostly, these MACs came from anoxic environments, marine sediments, but also hot springs or microbial mats. And doing this analysis again, eukaryotes appear to be placed more closely related to a particular group, the Heimdallarchaeota, that also turned out to have more of these, what we call eukaryotic signature proteins, proteins that were supposed to be exclusive of eukaryotes, but we have now shared with this archaea, and that most of all intervene in typical processes that are very extensive in eukaryotes, such as cytoskeletal cycling, endosomal cycling, trafficking, ubiquitin, etc. We have expanded today many more the number of genomes that are available for this type of archaea. Eukaryotes appear to branch really closer to this Heimdallarchaeota clade, Hodarchaeota. And by studying the functional potential of these different MACs, you can predict that these Ascararchaea are in general heterotrophs, and they can ferment small compounds, peptides, amino acids, and either release hydrogen or electrons, or sometimes scavenge that. And this strongly suggests that these organisms are interacting with syntrophic partners, with metabolic partners, bacteria or archaea, that would scavenge electrons or be donors for them, or hydrogen. And at the same time, by analyzing all these genomes, we have even enlarged the number of known eukaryotic signature proteins. We know now more than 500, mostly involved in this large category of intracellular trafficking, secretion, and vesicular transport. Not only these proteins are homologs, and of course the structure is very similar, but they can also complement the eukaryotic proteins. So in particular, this is the case for profilins, gelsolin, cofilin, which are players of the actin dynamic cytoskeleton, actually. And so this suggests that these archaea have an active dynamics of this cytoskeleton. So, if eukaryotes can be placed now within archaea using these very highly conserved markers, this implies that there were not 3 primary domains at the beginning, but only 2, and that eukaryotes are actually mergers. They derive from some form of symbiosis between archaea and bacteria. There was never a third proto-eukaryotic lineage independent from them. And this is actually coherent and congruent with the mixed heritage that we can observe in eukaryotes. Eukaryotes have archaeal-like informational processes, so all genes and proteins involved in DNA replication, repair, transcription, translation, but they have the bioenergetics more bacterial-like, via the mitochondria in particular, and also bacterial-like membrane phospholipids, which are actually very different from the ones present in archaea. So symbiosis has an extraordinary power, explanatory power in evolution, because when you have two different partners that establish this symbiosis, and this symbiosis becomes obligatory, that can happen when one partner gives transfer genes to the other partner and they are lost in the donor, then you have necessarily a change of constraints. You have genome redundancy, compartmentation, you have evolutionary flexibility, and one of the duplicate genes, for instance, can increase the evolutionary rate. That can lead to innovation, and all of that can result in the increasing complexity that we see at the level of eukaryotes. So what do we know about the different partners in this scheme, and about the evolutionary process that led to the origin of eukaryotes, and first of all to the last eukaryotic common ancestor, LICA? What do we know about this LICA? So we have known for a number of years now, using comparative genomics of eukaryotes, that this LICA, this ancestor, was already very complex and had more or less everything that extant eukaryotes have. They have a complex cytoskeleton, nucleus, endomembrane system, trafficking, ubiquitin, sex, etc. Something at least having more than 5,000, perhaps 10,000 genes as of today. What do we know about the prokaryotic ancestors of eukaryotes? We think that the host was at least related to an Asgard archaeon, perhaps a Heimdall archaeote-like archaeon. And about the alpha protobacterial ancestor of mitochondria, we don't yet have very clear neighbors, and actually recent studies to which we have participated and have incorporated a much better taxon sampling of alpha proteobacteria, including MAGs coming from very different environments that enrich the diversity, and using also models of sequence evolution that allow to accommodate for compositional biases and to cope with differences in evolutionary rate between different sites in an alignment and between branches in the tree of life, or bacteria in this case. This type of analysis show that mitochondria do not actually associate to any of the known groups of alpha protobacteria, so maybe there's another group to discover there, and in particular not to Rickettsiales, to which they were associated in the past, because Rickettsiales are, among other things, intracellular parasites. So more work to do here. When did eukaryotes appear? Here we have constraints from the fossil record on the one side, but also from molecular dating. So we know that the oldest bona fide eukaryotic-like fossils date back to 1.5 billion years old, and this is more or less coinciding with molecular dating saying that eukaryotes might have, the last eukaryotic common ancestor might have existed between 1.5 and 2 billion years old. There is another constraint, that is the ancestor of mitochondria was at least a facultative aerobe. It was able to respire oxygen, and this could only happen after the atmosphere was oxygenated. This is called the great oxidation event. As you know, the atmosphere of the early planet was anoxic, and it became enriched in oxygen by the activity of cyanobacteria, oxygenic photosynthesis. Okay, so this process more or less took place here, and then you had other events of endosymbiosis linked to the origin of the plastid and its dispersal in the eukaryotic tree. But the most open questions about the origin of eukaryotes relate to the how, which were the mechanisms involved in this process of symbiotic integration, and why, which were the selective forces that favor and trigger the process. And this is where most models diverge. There are many open questions. One of them is whether or not phagocytosis was actually needed. So many people think that, of course, prokaryotes don't have a very active cytoskeleton, and they don't have endomembranes, and then they lack phagocytosis. This is a classical idea. And so some models propose that some archaea would have developed phagocytosis over a long evolutionary scale by developing an active cytoskeleton and an endomembrane system. This is the phagocytosis in archaeon theory, and the Asgard idea would fit this model. Other models kind of propose a slow phagocytosis by proposing that the ancestor of mitochondria would be epibionts that would be little by little engulfed and surrounded by the secretions of the projections of the cytoplasm. So it's a sort of slow phagocytosis. But actually, we know that at least many prokaryotes do endomembranes, in particular many bacteria do endomembranes. You know very well the thylakoids in cyanobacteria. There are also many proteobacteria with endomembranes, methanotrophic alpha and gamma proteobacteria. And you have even bacteria that have endomembranes forming a nuclear-like compartment. It is not a true nucleus, but there is an inner compartment that is membranous. And furthermore, recently, a Japanese team described true phagocytosis using a different mechanism in bacteria. So there are some plactomycete bacteria that can engulf, and you can visualize that, and there are videos, so you can see that. And these bacteria prey on other bacteria, they engulf them. So there is phagocytosis, at least in bacteria, maybe not in archaea. We don't know. So, and another question is relates to the nature of the symbiosis that was at the origin of eukaryotes. And we think that this interaction was based on syntrophy, on metabolic exchange, as you said. And actually, this is further supported by the culturing of the first and unique so far Asgard archaeon. So Asgard archaea were predicted just from Max, but now we have a culture, and this organism has this type of protrusions, so suggesting that indeed there is a cytoskeleton pushing these structures. This organism is an obligate syntroph. It needs to live either with the methanogenic archaeon or with the delta sulfate-reducing delta proteobacterium that are proton scavengers. And so this, together with the fact that from archaeal max you infer that all Asgards are proton or electron donors or scavengers, actually suggests that these archaea interact with others metabolically in natural environments. And that also constrains the types of environments where you can find this type of archaea and where likely eukaryogenesis took place. These are redox transition environments, where, and mostly anoxic actually, where you might have a little oxygen at the surface, but then you have a gradient of different donors, electron donors and acceptors. And under these conditions, where you don't have a strong oxidant like oxygen, in these particular environments, you have only weak electron donors and acceptors, you actually need, there's a syntrophy is favored, you actually need a sink for your electrons or your hydrogen or other compounds in order for your metabolism to proceed. So syntrophy is actually a dominant force in this type of ecosystems. So, and it is in this context and based on microbial ecology data that we propose in the 1980s our first syntrophy models that we have now updated to accommodate that Asgard archaea are related to eukaryotes. So we now propose that eukaryotes within this syntrophy model evolved from the endosymbiosis of an Asgard archaeon within a delta proteobacterium that was a sulfate reducing. So this archaeon was fermenting amino acids liberating hydrogen that was used for sulfate reduction by the host, and a second symbiosis and then endosymbiosis happened with the ancestor of mitochondria, which was sulfur oxidizing and recycled sulfur in disassembly. And these interactions are known in nature, so these are real. That they were or not at the origin of eukaryotes is another thing, but it is not unlikely. And during the process of integration, many things happen, including the development of an endomembrane system that had a secretory purpose at the beginning to actually transport the proteins that were starting to be synthesized. The synthesis of these proteins was centralized by this consortium, by the archaeon, and they needed to be exported to the periplasm for degradation. I'm not going to enter into the detail. There is a process of integration related with genome evolution as well and gene transfer. This is just one model among others. The rest of the models of eukaryogenesis to date, most of them involve a host that is an archaeon and not a bacterium, and with only two partners, the ancestor of mitochondria and the Ascar archaeon. There is one model involving sulfate-reducing bacterium, but it went away in a second step. And some models include more symbionts. There are many open questions about the origin of eukaryotes. There are questions about the nucleus. Actually, most models of eukaryogenesis do not address the origin of the nucleus. In our model, the syntrophy hypothesis, the nucleus derives from the Ascar endosymbiont. But in other models, there's simply no proposal at all. There are some ideas about the role of introns in that. But this is the natural, selective forces and the mechanisms leading to the origin of the nucleus is unclear in many models. Another problem relates to the nature of membrane phospholipids. We know that eukaryotes have bacterial-like membranes, but most models propose an archaeal host. So in those models need to propose a transition from archaeal-like to bacterial-like phospholipids that is not easy to explain, because if your membranes are well adapted to interact with the rest of the world, and including the proteome that is sitting in those membranes, why do you need to change them all of a sudden? So there are problems about that. Were there other bacterial partners, and was the mitochondria an early or a late acquisition? And here also models diverge, because for some models, such as the hydrogen hypothesis, the acquisition of the mitochondria is the first step. It was somehow the cause of eukaryogenesis, whereas in other models, such as the phagocytosis archaeon model and related models, the mitochondrion was a late event. So there was somehow the consequence of an evolutionary process of eukaryogenesis that had already been triggered in the past. Some hints of answer are already available. In particular, with respect to this question, mitochondria early or late, in particular, this comes from work by Alex Pittis and Tony Gabaldon, who studied genes of prokaryotic origin in the last eukaryotic common ancestor. There are 1,000 genes of that kind, and measured the stem branch length separating in phylogenetic trees these genes of prokaryotic origins and their prokaryotic relatives. And by doing that, they noticed that branches uniting eukaryotes with alpha protobacteria were much shorter than those linking eukaryotic genes with those of other bacteria or with archaea, suggesting that the archaea were older and the ancestor of mitochondria was more recent. And in addition, they did the same for the types of processes these genes were involved in, and again, mitochondria and plastid-related functions appear to be more recent, had shorter branches as compared to those involved in nuclear and endomembrane processes. The thing here, or the interesting observation in this study, is that from the around 1,000 genes so far of prokaryotic origin present in LECA, Only a fraction is of archaeal origin. Most of them are bacterial in origin, but when you look at their distribution, only a small fraction of them are related to alpha proteobacteria. The rest are related to other groups of bacteria. What does it mean? How did these genes arrive there? One possibility is that they arrive by horizontal gene transfer to the Asgard ancestor of eukaryotes, or to some intermediate eukaryotic ancestor. Or the possibility is that there were several endosymbionts or several symbionts that were replacing one and the other, and each one led some portion of genes. So there are many open questions there. Of course, the two are possible, a mix of the horizontal gene transfer and a mix of endosymbiotic gene transfer. And this brings me back to the question of which were, or how many prokaryotic partners were involved in the origin of eukaryotes. And here I want to mention that many people think that if you only have that, having only two partners is more parsimonious, and so that should be favored, and that's what most models do. But when you go to nature, you realize that evolution does not necessarily follow the most parsimonious way. There's a lot of contingency out there, that multiple symbioses are very frequent, and successive symbiosis as well, symbionts that replace other symbionts. So with all of that, I want to simply want to convey a few take-home messages. So the discovery of Asgard archaea, thanks to the exploration of the microbial world out there, have actually favored now symbiogenetic models of eukaryogenesis that are based on metabolic symbiosis, on syntrophy. We need a lot to explore the closest and to understand which are the closest prokaryotic lineages to eukaryotes, thanks to phylogenomic analysis and study their biology and their ecology. And this will help eventually to discriminate and refine plausible models of eukaryogenesis. And models of eukaryogenesis need now to depict detailed plausible processes that account for observed patterns in a realistic ecological context. They need to take all the data into account. We need the details. And so with that, I thank you very much for your attention. I thank particularly David Moreira, who shared with me many of these archaea, long-term collaborator, and my team at Orsay, now Gilles Rivet. And of course, Esquerrik asko, thank you very much for your attention. Welcome, and question is welcome. Thank you.
Thank you very much, Puri. There are questions for Puri?
Thank you. It was a fascinating talk. So you talk about symbiogenesis being quite a frequently occurring thing. Is it unlikely for eukaryogenesis to have happened multiple times then? Is our understanding significant enough that we can say statistically that the event only happened once?
Yeah, we can say that the event only happened once because eukaryotes are monophyletic. So we can trace a last eukaryotic common ancestor. There is a single organism. If we had multiple origins, we would have polyphyletic eukaryotes. So that's the simplest answer. Of course, these processes of symbiosis are happening all the time. What is difficult is to make these symbiosis integrate. Usually, many of these symbioses, where you have the symbionts tend to lose genes, etc., they tend to disintegrate. Here, we have an integrative process that persisted over time, and that's the unique thing.
So, continuing, I just had some curiosity, what your thoughts are. Because sometimes you hear about the origins of life coming from someplace else. What do you really think? Tell us a little bit about that. What do you think?
Panspermia.
Yeah.
I think, the origin of life is a very interesting question, and for sure. In very complex and interdisciplinary, I think that panspermia is not really something very solid. We could imagine transportation of life in some type of meteorite, very well protected, etc., from eventually Mars to the Earth, but that's all. And even that is extremely difficult, but not from elsewhere. So panspermia is actually a way of transporting the problem of the origin of life elsewhere. Most likely, life originated on Earth. We don't know if it happened in any other planet. We have no evidence for that for the moment, even if there's a lot of wishful thinking, but we'll see. So yeah, it's not a solution, panspermia.
Yes.
Thank you, Professor, for your presentation. It was amazing, actually. And I have a couple of questions. The first of this one is like, I wanted to know if you could pinpoint the location where this first like LECA organism appear, like the ecosystem itself.
Mm-hmm.
Is that possible? I mean, if there is only one.
It is difficult, of course. What I mentioned is most likely eukaryogenesis involved in this type of redox transition environments, places where you have different electron donors and acceptors, but the presence of oxygen not too far, because you need the ancestor of mitochondria was most likely a facultative aerobe, so it had to have access to oxygen sometime, but also was respiring other things for most likely. And so yes, we think of an environment where you have this type of transition. This could have been sediments. Most Asgard archaea are found in sediments. There might be other places where you have suboxic conditions, but then of course there are many types of environments. So microbial mass, sediments, eventually, yeah, freshwater systems where you have, or marine systems where you have low oxygen conditions.
Okay, perfect, because this leads to my second question. So I was wondering that maybe this symbiont approach could be dealt with the idea of a mixotrophy in between, like, you know, a facultative heterotrophy at some point in this kind of ecosystems.
But yes, that's absolutely possible, and these type of symbioses are so widespread. I mean, in nature, they are not the endosymbiosis, but the natural, this type of symbiosis that are seen for Asgards, but also for many other microbes, in particular for delta protobacteria and methanogens, methanogenic archaea. They are involved in lots of these type of interactions. And in all these communities intervene in the final degradation of the organic matter that comes down into sediments or soil. And there, yes, you have different types of electron donors and acceptors, and this type of syntrophies because you need a sink. You need someone pulling your reactions down. I don't know if I'm answering your question or not. But this can be any, I mean, these environments are so widespread in the planet, even today, that, yeah, you cannot say it happened in a geographic location, or was it really in a plankton-like, if it was a plankton-like, it could have been associated to particles, where you have also these redox gradients. But we don't know for sure.
Thank you so much.
So do you think that if we continue looking into those areas, these niches, anoxic areas, or so, we may find some intermediate?
I think that's difficult because what we know, people are looking for that, and it's a very interesting approach because we can learn about how symbiosis happens and whether you can have more or less integrative process. I mean, we know now many protists have endosymbionts, and it may be actually the rule that most protists out there have endosymbionts, different bacterial endosymbionts.
It's more the rule than the exception, right?
It's more the rule than the exception, right. Absolutely, including in between prokaryotes. But these things are not that easy to study because most symbioses are facultative. They are not obligatory, and so effectively, if we change the conditions, they can simply do their own thing. But yeah, we have here a case of a symbiosis that integrated over time, that was obligatory, became obligatory at some point, and yeah, persisted and amplified.
Any more questions for Puri? Well, we'll thank Puri for
Thank
this
you very
great
much.
talk. We are pretty much on time, a bit late, but not so much. We're going to take a break now. I can't remember how much. It's about half an hour. We still have, yeah, half an hour a break. There is some coffee in the Grand Salle and something else for some people probably. I'm not sure there is enough coffee for everybody, but anyway, you can try. There's going to be something in the Grand Salle, okay? And so if it is 11:20 now, hey guys, wait. 11:20, we should be here by 10 of 12 sharp, okay? 10 of 12 sharp.